This is the question. Okay, it's an N of 1. Okay, so the lung issue is a completely different issue. What we did in one patient carrying that mutation in an LRSC4, that we had difficulties in controlling background inflammation. Since an LRC4 is very important in gut epithelial cells, and alatine is produced in gut epithelial cells, we thought that it was important in response to pathogens that we were isolating from the stools of the patient, and we decided to do a fecal transplantation, which means that you can change the entire intestinal microbiota by essentially using feces from a normal donor. And this worked out impressively nice. It's an N of 1. We don't know what it means, but it was presented at EULAR, and Alexi picked it up.
And I think there is certainly a connection, I think, between the gut microbiota and sort of systemic inflammation, but particularly within MAS. I think the mice with the NLRC4, it seems like it's the gut cells that are making a lot of that IL-18. And there was a paper from looking at one of those mouse models that Fabrizio mentioned of MAS, where if you give those mice, if you alter their gut microbiota, they don't get MAS anymore. So I think there is a strong connection which we completely don't understand between what's happening in the gut and how that regulates your systemic immunity.
Rashmi and I, we had multiple discussions about this particular topic, and what we believe. Rashmi and I, we had multiple discussions about this specific topic, and what we believe might be happening is, remember Grant showed a slide showing that if you look at the gene expression in the lungs, in the inflamed lungs, we see a very, very strong interferon gamma induced response. So we think that's what drives or sustains inflammation in the lungs. Now, many infections, when we get those infections, we actually use the same type of immune response to fight those infections. So the question is whether we have this underlying inflammation in the lungs, and then the immune response that is induced by those infections synergizes with that already ongoing inflammatory activity and makes it worse. So we don't think it's a causal effect, but it's something that potentially can synergize with already ongoing inflammation in the lungs. And for that reason, I think we do need to understand a little bit more about what kind of bugs actually live in the lungs and what may promote that particular immune response.
I was going to say exactly, we need to understand what's going on in the lung. We need to study the microbiota of the lung. We see. So I think it was pretty clear by looking at your patient yesterday. Some of the kids, they do well. We don't understand why they do well. We think it's treatment, but it may not be treatment. It may just be a way to find a new equilibrium with the microbiota, which I think is important.
Just want to say, wait for the microphone so that we can make sure that all your voice gets on the video.
Hi, I have a daughter who just was diagnosed 7 months ago, and she was 7. She had SJIA MAS, and her IL-18 was 321,681, and her ferritin was 58,620. So I'm wondering, I'm not really sure your name, I don't remember, if we should look at something. I know she did the study, but should we look at something if her IL-18 is still high to block that? I know she's not on it. She's on cyclosporine and she's on Kineret, and she's doing amazing. But I'm wondering, I was listening to you about the IL-18 blockers.
This is a very difficult question. So we don't know. IL-18 has been used, as far as we know, in one patient with an LRC4-induced disease, is being tested in LRC4-induced disease. And so this is what we know from a perspective of clinical response in patients. We certainly have plenty of data. We all measure alatine, and a lot of people do measure alatine. We have plenty of data showing that patients with systemic JIA have very high level of alatine, particularly when they have mass and if they have interstitial lung disease, which doesn't mean that everybody who has high alatine gets MAS and gets interstitial lung disease. Simply means that alatine is there, is part of the hyper response of patients with systemic JIA, and it's not necessarily a bad guy. What I'm trying to say is that we have seen patients, and I'm sure that you have done the same. We have seen patients with very high level of interleukin 18 in the range of hundreds of thousands, like your daughter, who are perfectly fine. And even if you treat a patient effectively with anakinra and sometimes with anakinra only, alatine takes forever to go down. And by forever, I mean take months or years to go back to a reasonable level. So it's not necessarily bad, it's just there. And the biological basis for being there and not necessarily being bad is that it is a cofactor. That alone does do very little. Okay? Is that an answer?
Yep, that's wonderful. Thank you.
You were saying something.
I think you pretty much hit what I was gonna make the comment that high alatine doesn't mean that the child is sick or that their treatment isn't working. We have Dr. Yazin and Dr. Grau and myself have a paper in press, which one of the things that it says is there's actually no relationship between how long a patient has been in remission on medication and their alatine levels. It's completely random. And that some of the data from the trials of canakinumab showed that there was no relationship between who responded great to the drug and their alatine level falling. So it's hard to understand how to use that to think about if treatment's working or not.
I would like to make one more comment. So both Grant and Fabrizio showed a slide about the IL-18 biology. So we have the cytokine that is produced, and then we have this the IL-18 naturally occurring IL-18 binding protein that has the ability to neutralize IL-18. My feeling is that your child perhaps is producing enough of that IL-18 binding protein that is completely neutralizes the activity of IL-18. So you were asking about the drug that would neutralize IL-18. It's exactly the same molecule, but I think your daughter is producing enough of that molecule, so there is no need for additional treatment to neutralize this cytokine.
What, if any, correlations are there between the age of onset and the severity of the SJIA and the potential of complications such as HLH and MAS?
I mean, the only one that we've found is this relationship with lung disease, that it does seem to be children who are diagnosed at a younger age, although it's certainly not exclusively. I don't think there's any association with MAS, maybe also.
All times, Grant is too young.
Thank you for that.
You're very welcome. In the old times, yes, there is. There is a very nice paper from Argentina showing in several dozens of patients with onset before 2 years of age that they have a higher incidence of MAS, a more severe course, and therefore this is a group of children that also gets more frequently lung disease. So I think these are the most severe one, the one we should pay the greatest attention to in terms of early diagnosis, early treatment, and possibly, I hate to use this word, but aggressive treatments so that we can get control of the disease as soon as possible.
Hi, thank you guys so much for your time. So just about the IL-18, we're from Canada, and the hospital that my son is being seen at doesn't test, or at least hasn't. He's only been 2 years diagnosis, IL-18 levels, or any any interleukin levels. Is there any value in knowing that? Like, are we looking at the future? Well, right now he's controlled, but I know they can relapse, and so much is unknown. Is there any value in going, okay, well, we need to know these levels for future reference, or is there anything that it will indicate that, okay, we need to be looking for lung disease or any other complications in the future?
I don't think you need to worry about that. As long as the disease is controlled. How old is your son?
He's 14.
14. I would not worry about that.
Have there been any studies done related to prenatal or neonatal conditions? I had a neonatal alloimmune issue with my son. Has there been any research regarding pregnancy and autoimmune, or?
Well, certainly I'm not aware of any studies like that, but I would like to stress the point that we believe that the adaptive immunity, the immunity that would be involved in those kind of immune responses, is probably functioning just fine in patients with systemic juvenile idiopathic arthritis. The issue here is the innate immune responses. And I don't think that what you're referring to has much to do with that specific part of the immunity.
Microphones. I hate microphones. Hi, thank you. I just have a quick question on siblings. Are we being more proactive in testing siblings of SJIA patients to see if they are carriers or not carriers, are going to show the IL-18? Because my daughter is actually a twin and nobody's tested the twin yet. So yes, we're participating in the study today. I'm just wondering, is there proactivity in testing other siblings, not necessarily twins or not? Or?
If you look at the genetic studies in systemic juvenile idiopathic arthritis in general, those studies, the results have been quite disappointing. We really have difficulty finding variants, genetic variants that would explain the disease as a whole. Perhaps some components of the disease, but not the disease itself. And we believe that actually the genetic component is probably very weak in this condition compared to other rheumatic diseases. In addition, I can tell you that at some point here in Cincinnati, we collected samples and clinical data on approximately 270 patients with systemic juvenile idiopathic arthritis. In that collection of samples and clinical data, only 2 had siblings with a somewhat similar disease. And when eventually, more recently, Grant did some whole exome sequencing on those 2 families, they actually happened to have a completely different disease. It was hyper-IgD syndrome. So my point is that, so if we remove these 2 patients of that large collection of families and samples from the families of a patient with systemic JIA, we did not have a single case, a single family where another sibling was affected. So for that reason, just for that reason, I don't think you have reasons to worry about your siblings.
One second.
I just know there's another set of twins in Tennessee where I'm from that they both have.
Yes,
so
and I
that's
suspect
what I
that
was
it might be a different disease that we just did not diagnose properly.
Okay.
So my question is kind of on the name, systemic JIA, and the reclassification process from autoimmune to autoinflammatory, and then what is the progress on that, and what is the progress on the research into, I know last year was talked about the subsets, kids with arthritis may be a different subset than the kids with the systemic features. So I was just wondering what the progress was on that front, if there's any.
You're perfectly right. This is on classification. It's a major issue, and the guilty ones are sitting here, are the physicians. So we have really been messing up on this. George Frederick Steele described the disease in children, which is what we call now systemic JIA. We group this disease together with JIA, and JIA are autoimmune diseases, and they are not autoinflammatory. They have a completely different pictures, a completely different genetic background, a completely different treatment approach. So it's a completely different ball game, and it's wrong to classify systemic JIA together with JIA. Then the issue is that the adults become calling adult onset Steele disease what is actually JIA, systemic JIA, and it got even worse and more complicated. And moreover, we applied a very arbitrary is a euphemism here, cutoff of 16 years. Why? This is the same disease occurring throughout the life, being certainly driven autoinflammatory, characterized by very good response to IL-1 and IL-6 inhibition in most of the patients, very high hyper innate immunity, and high risk of MAS. And it's always the same stuff in adults and kids. So we should change it. And the most amazing things that have happened for once is that the regulatory agencies, the FDA and the EMEA, have bought the idea that this is the same disease and approved the drug and changed the labels of the drug and approved the drug in both adults and kids, essentially before us could make up our minds. And and it's embarrassing, yes it is. And we can't come up with an idea because medicine is going away from eponyms. They don't like to name diseases with the name of the doctor who discovered it or described it. So Steele's disease, it's outdated and we don't know how to call it. That's the point. So there is no progress that I can mention. And yes, there is heterogeneity. There is heterogeneity, but it's a continuum. Okay, some kids have zero arthritis, but it's really zero. They have very little arthritis. Some of them have a lot of arthritis. Some of them have clinical overt hyperacute MAS. Some of them have less acute MAS. Some of them have subclinical MAS. So I think it's really a spectrum of, of one disease. But this is my idea.
Can I ask a follow-up question to that? And I can't believe I'm raising this with you because we've discussed this many times. In this naming process, do patient organizations get a say? This is something that we talk about very often in the group and our frustrations with it and how it impacts everyday life for these kids. And I always say, let's just be pragmatic, there's nothing we can do here, let's focus on the problems we can solve. But when these naming decisions happen, how about including, how do we get included in that conversation?
There is no such a naming process. There is no such process ongoing. There was one attempt made by a group of eminent people, I was not part of it, that agreed, that did not even agreed upon naming, taking SJIA away from JIA, which I think was completely wrong. But my opinion doesn't matter. There were 15 people around the table. They did not reach consensus on getting it out, and therefore it stays in. But this is not the way it should be done. I agree with you. This should be done in a completely different ways, with different components, taking into account the biology. This is not only clinical features, it's biologically driven. And we hope that the biology becomes sufficiently strong that the classification of patient is not anymore based on presence of fever that has to be quotidian for at least X number of days, and fever and rash and whatever is in there, which doesn't make any sense, but it's based on a set of biomarkers. This is 2020. I think we should aim for that. I'm too old to see it.
I just want to make a short comment. I thought that was a great question. And what has happened in 1 year? That was very interesting, the perspective that we expect a lot more to be known in 1 year. The pace of discovery has just accelerated, kind of like Dr. Schuler's graph when everything was going up, with the number of patients discovered with it. our detection, the different tests we have all changed very rapidly. And I think we're all trying to move together as a field to really put our hands around it, our heads around it. And a lot of people are asking questions, and patients are asking questions, researchers are asking questions, clinicians are asking questions. So I think there's going to be continual change and discovery. I'm up here, I'm a clinician, Essie Morgan. And, Rashmi said, oh, you could go on the panel. And we always have this imposter syndrome, for the SJIA expert as clinicians, but we do see patients come to us. And so it behooves us to become as expert as we can be, not being on the front lines in the lab, et cetera. But we're learning a lot of this at the same pace that you are. And so I really appreciate this conference, too, that is sharing sort of cutting-edge research. But clinicians also have to keep on top of it. And your question about should we be sending IL-18, we're also learning this ourselves. We all, once it became available, a bunch of us started sending it. And then we realized, gosh, this is a really expensive test. And it's high or it's low, and the patient looks good, and they look good, and, how do we use it? So we said, let's take a pause, and let's stop sending it on everybody, but let's study it a little better and understand how to send it, before we make it like, oh, that's quality of care, we need to send it on everybody. No, let's understand first. So that was just the general observation.
I would say Dr. Morgan sells herself short by saying she's just a clinician, but she also helps run our division's quality improvement efforts as well as them nationally, and is working with us to help us think about these exact things, is how can we measure the quality of care we're providing for our patients with systemic GIA as well as other autoinflammatory diseases, and bring these new discoveries not just to the bedside, but in the best possible way that's going to actually impact the quality of care instead of just give us more data that we don't know what to do with.
Hi. So I was just looking at your slides that you had for blocking interferon gamma, and my question was, interferon gamma does a lot with the innate immune system. Do we know what else is being used to fight these kids' opportunistic infections when we block interferon gamma?
I think I should answer the other way your question. We know what happens in humans that are deficient for interferon gamma activities. And we know this from two sets of individuals. There are individuals who lack the interferon gamma receptors, and therefore they have zero interferon gamma effects on the innate immune system. They are fine unless they get infected with mycobacteria of various species and with some other very rare intracellular pathogens. We also know from patients who developed autoantibodies to interferon gamma, that's a very weird phenomenon, but it does happen, they get again into trouble if they get mycobacterial infections, and they have a little bit more of herpes zoster reactivations. Okay, but other than that, they're fine. But this is something that is very well known in the cytokine field. If you inhibit one single cytokine, the hole you make in the net of the responses is not that big. It's a small hole. So somebody gets in this case mycobacteria, okay? But not much else. If an interferon gamma receptor deficient human gets measles, which is a very dangerous disease, he's fine. So what we do in the study is that we screen for these rare infections that are potentially dangerous before, and if you have one of these active infections, this is actually an exclusion criterion. And then we screen during the study, and we watch for this to happen, okay? There was one infection in the primary HLH study with Histoplasma capsulatum. I don't even know what that is, but it doesn't really matter. It's one of those infections that are potentially favored by defective interferon gamma. So the drug was withdrawn, but was still there because it stays in the blood for a while, and the patient was treatable with the standard treatment, and the infection was resolved. This is all we know. But I think on this side, it's a very safe approach because we have data from Mother Nature, which are the strongest data of all.
Got time for 2 more questions.
Hi. We have, our son has SJIA, was just diagnosed about 2.5 months ago, and he's been on anakinra. And we're trying to get him switched to Alaris, and have run into problems with the insurance company. But my question is the following. Comparing Alaris versus anakinra, is there any understanding of whether one would make a patient more susceptible to infection than the other?
If you look at the published data, I don't think we see that much difference in terms of rate of infections. I think there are a couple of questions. So the first question is, when you use these drugs, does that treatment put you at a higher risk to get infection, correct?
Right, right.
And then the second question is, if you do get the infection, does it take the same amount of antibiotics to treat this infection? So the available data does not suggest significant difference between the two biologics. However, I would say we have quite extensive safety data for canakinumab, because there was a large clinical trial with a long-term extension phase, about, I would say, 700 patient years or something like that. I don't think we have comparable databases for anakinra, so the numbers that we have for anakinra are probably based on smaller studies. But in general, again, if you look at the literature, sort of if you think about healthy kids, if you follow about 100 healthy kids, I would say probably 1 or 2 would get infection within 1 year that would require antibiotics. With both canakinumab and anakinra, I think the risk increases probably 7-10 times. But still, when these kids get this infection, the treatment is still the same. I don't think they require longer treatment with antibiotics.
Okay, thank you.
Thank you so much. I think it's time for lunch, unfortunately, and any parents that have their kids in childcare, you do need to go up and pick them up, and they're getting kind of restless and hungry right now. Thank you.
So are we.